Hello everyone. We are going to share and explain about the synthesis of cholesterol. So, let's get started. What is cholesterol? Cholesterol is a waxy and an oil-based substance. It is found in every cell of the body. Our body needs cholesterol to generate vitamin D, produce hormones, and also substances that help body to digest the foods. Cholesterol is a crucial substance that the body generates, and it is also found in the foods that we consume. Cholesterol does not mix with the blood, which is water-based, and it travels around the body via lipoproteins. Did you know? Nearly every tissue within the body is able to synthesize cholesterol from AcetylCoA. The liver represents approximately 20% of the endogenous cholesterol. The intestine is probably the most active among the extrahepatic tissues, that are responsible for the remaining 80% in synthesizing the cholesterol. In the production rate of the cholesterol, it includes both absorbed cholesterol, and cholesterol that are being synthesized endogenously. The recommended dietary intake of cholesterol is not more than 300 mg per day. Average intake of cholesterol is estimated to be about 600 mg per day, which just about half is taken up. Hence, endogenous synthesis of the cholesterol accounts for over 2/3 of the daily total. Now, we are achieving the part where cholesterol is being synthesized. 5 major steps in cholesterol synthesis. 1. AcetylCoA converted to HMGCoA. 2. HMGCoA to mevalonate. 3. Mevalonate to isoprene-based molecules. 4. IPP and DMAP to squalene. And 5. Squalene to cholesterol. Step 1. AcetylCoA converted to HMGCoA. 2-acetyl-CoA condensed by thiolase to form 1-acetoacetyl-CoA. This step release 1-CoA. Then, the acetoacetyl-CoA undergoes addition of 1-acetyl-CoA by HMG-CoA synthase to form HMG-CoA. This step releases another 1-CoA. Step 2, HMG-CoA, converted to mevalonate. HMG-CoA catalyzed by HMG-CoA reductase to form mevalonic acid. In this step, 2 NADPH and 1 ion hydrogen are utilized and release as 2 NADP+ and 1-CoA. Then, mevalonic acid undergoes 2 times hydrolyzation by kinase to form mevalonate. Step 3, mevalonate, converted to isoprene-based molecules. Mevalonate undergoes decarboxylation by decarboxylase to form IPP. This step utilizes 1 ATP to remove 1 carbon atom in the mevalonate in the form of carbon dioxide. This causes release of 1 ADP, 1 phosphate group and 1 carbon dioxide. IPP then undergoes isomerization by isomerase, forming DMAP. Step 4. IPP converted to squalene. Squalene consists of 30 carbon. Synthesis of squalene requires 6 forms of isopentenyl pyrophosphate and involves 3 steps. The sequences of the reaction is right below. This stage starts with isomerization of isopentenyl pyrophosphate, IPP, into dimethyl pyrophosphate, DMAP. Let's move on. Firstly, 1 molecule of IPP and 1 molecule of DMAP are condensed, forming geranyl pyrophosphate, which consists of two isoprenes. This reaction is catalyzed by cis-prenyltransferase and eliminate one molecule of pyrophosphate. The geranyl pyrophosphate, or GPP, consists of 10 carbons with 2 molecules of isoprenes. Secondly, the GPP is condensed with another one of IPP molecule, forming farnyl pyrophosphate. This reaction is catalyzed by cis-prenyltransferase and eliminate one molecule of pyrophosphate. The farnyl pyrophosphate, or FPP, consists of 15 carbons with 3 molecules of isoprenes. Lastly, the 2 molecule of FPP are condensed into squalene by squalene synthase. The synthesis of squalene is catalyzed by the NADPH requiring enzyme and eliminate 2 molecules of pyrophosphate. The squalene produced consists of 30 carbons. We are achieving the last step of the synthesis of the cholesterol, where squalene converted to cholesterol. In order for squalene to be converted to cholesterol, squalene undergoes a 2-step cyclization for linosterol to be generated. In the first reaction in this 2-step cyclization, enzyme squalene epoxidase, which is also called squalene monooxygenase, catalyze this reaction. Enzyme squalene epoxidase uses NADPH as a cofactor to introduce molecular oxygen as an epoxide at the 2,3 position of the squalene. This forms the intermediate 2,3-oxidosqualate. Next, during the second step in this 2-step cyclization, the epoxide intermediate is then converted to linosterol through the action of an enzyme called linosterol synthase. The process where linosterol is converted to cholesterol does take place in the membranes of endoplasmic reticulum, and it occurs through a sequence of 19 supplementary reactions. These 19 reaction steps are catalyzed by 9 different enzymes, which are either located at the endoplasmic reticulum or at peroxisomes. Lastly, the terminal reaction in cholesterol biosynthesis, where linosterol is converted to cholesterol, is catalyzed by the enzyme 7-dehydrocholesterol reductase. This enzyme is encoded by the DHCR7 gene.